Dermal tissues are located on the surface of all plant organs. They primarily perform a protective function, shielding plants from mechanical damage, microbial invasion, sharp temperature fluctuations, and excessive evaporation. At the root tips, there is also a specialized primary dermal tissue with root hairs called epiblema, which combines the functions of protection and absorption. However, it is short-lived, and as the root grows, it is replaced by the outer part of the cortex, the exodermis. Dermal tissues include the epidermis, periderm, and erythrodome. Primary dermal tissue, the epidermis. The primary dermal tissue, the skin or epidermis, covers the surface of leaves and young green shoots. The epidermis protects plants from excessive evaporation and dehydration, sunburn and hypothermia, as well as the penetration of parasites and pathogenic fungal spores. The epidermis is formed from the primary meristem, the protoderm, and usually consists of a single layer of living, tightly packed cells that lack chloroplasts. Chlorophyll is found only in epidermal cells of ferns and certain shade-loving or aquatic flowering plants. The outer walls of the cells may be thickened and impregnated with a waxy substance called cutin. Due to this impregnation, the surface of the epidermal cells is covered by a film, the cuticle, and sometimes by a wax coating of varying thickness, providing additional protection. The thickness of the cuticle and wax coating depends on the plant species and its habitat. They are thickest in inhabitants of arid regions. In tropical forests, the cuticle and wax coating make leaves water repellent and prevent the attachment of small epiphytes. In the delicate leaves of shade-loving plants and the submerged leaves of hydrophytes, the cuticle is absent. To protect against overheating and excessive evaporation, the surface of the epidermis in many plants features various scales, glands, and hairs, outgrowths of epidermal cells known as trichomes. In terms of shape, size, color, and distribution, they can vary widely. Long or short, oppressed or spreading, straight, wavy or curly, white, silvery, yellowish or red, 2-3-parted or even branched. Epidermal cells in different plant groups may have rectangular or wavy outlines, or they may be elongated, as seen in monocots. Wavy cell walls help the cells interlock firmly. Due to the cuticle and tight interlocking, epidermal cells are almost impermeable to water and gases. In the epidermis of leaves and green stems, there are microscopic openings called stomata, which fulfill one of the most important functions of dermal tissues, regulating the evaporation of water or transpiration and gas exchange. Stomata appeared in higher spore plants after they moved onto land and have undergone a long evolutionary path, reflecting the mechanisms of exchange with the environment. Transpiration is a necessary condition for the creation and maintenance of the flow of water and dissolved minerals absorbed from the soil. It prevents leaves from overheating, maintains their tissues in a state of partial water saturation, and thereby helps maintain the suction force of the cells at a certain level. Through specialized water stomata, hydrathodes, located on the serrations and tips of leaves, the release of liquid water droplets occurs during humid periods. This phenomena is particularly characteristic of tropical rainforest plants. In addition to stomatal transpiration and guttation, evaporation also occurs across the entire leaf surface, cuticular transpiration. Its volume accounts for an average of up to 15% of stomatal transpiration and depends on the stage of leaf development, the composition and thickness of the cuticle, the time of day, and the ecological needs of the plant. High cuticular transpiration is characteristic of plants in humid habitats. In succulents and xerophytes, it is practically non-existent. The secondary dermal tissue, the periderm, replaces the epidermis in perennial plants. It is formed by the secondary meristem, the phylogen, whose cells divide and differentiate, producing cork outwardly and a layer of living storage parenchymal cells inwardly. Cork cells die and fill with air, resins or tannins. The walls of cork cells are impregnated with a fat-like substance called suberin, making them impermeable to water and air. The multilayered cork forms a kind of protective sheath for the stem, reliably protecting the plant from unfavorable environmental influences. As the cork forms, the green color of the shoots turns brown. Protected by the periderm from drying out, first-year shoots are able to withstand winter frosts. Cork also frequently develops at sites of damage, such as on tree trunks along the edges of frost cracks. Cork is multilayered, with its thickness in different plants ranging from a few millimeters to several centimeters. Particularly powerful cork develops in certain trees, such as the Amur cork tree and the cork oak. For gas exchange and transpiration of the living tissues lying beneath the cork, specialized structures called lenticels exist, ruptures filled with loosely arranged cells. The cork, phylogen, and phyloderm together constitute the periderm. Under the phyloderm of many perennial plants, a new layer of phylogen can arise and begin forming a new periderm, pushing the older layers outward. This process may repeat several times. As a result of the formation of new periderms, the outer tissues become isolated from the central part of the stem and die. Consequently, a complex of dead periderms accumulates outside the active phylogen, which is called the rhiziodome. Over time, rhiziodome layers can become quick thick, up to several centimeters, developing and even cracking. The rhiziodome protects not only the trunks and branches of trees, but also their roots.